Good day, everybody. It's a big pleasure for me to be here. I want to present you our new software tool, SeqWord Motif Mapper, to work with, to analyze bacterial epigenetic modifications. What is included into epigenetic modifications? This is epigenetics basically is the study of heritable and stable changes in phenotype which do not involve changes to the underlying DNA sequences. First, which comes to mind is, of course, methylation, DNA methylation, which is common for eukaryotes and prokaryotes, but it's involved more modifications like oxidation of nucleotides, like guanine oxidations under stress conditions. It's involved physical arrangement of chromosome, which makes some genes more accessible than others. It's involved some modifications of proteins and mRNA, regulatory RNA, and even inheritance of cytoplasm composition from mother cells to daughter cells, all involve this is epigenetics. But okay, now our program now focus on modifications, chemical modifications of nucleotides, like methylation. What role of methylation in bacteria? It's not the same like in eukaryotes. It's a kind of immunity of bacteria. We have clusters of restriction modification system, which built of 2 or 3 genes. They methylate nucleotides at specific motifs. So identify motifs in DNA. This is known like canonical motifs. And methylate here. Why it's important? Because if foreign DNA comes here, it's not methylated, and the restriction enzyme, cognated restriction enzyme, identifies them and cleave at these positions. So methylation prevents DNA from restriction enzyme. But many often methyltransferases can be found in chromosomes which are not associated with restriction modification system. Often they methylate DNA at one strand or at both strands, and the role may be in regulation of gene regulation. Very little is known at the moment which roles may play these modifications, and our idea was to provide tools to analyze patterns of modified nucleotides. What we can create? Program can create such graphs. You can see a circular graph with locations of modified nucleotides at both strands. It can create this dot plot, 2-dimensional graph to compare coverage and scores associated with each modified nucleotides, how likely this nucleotide is modified. And very important, we have statistical plot, which represents for us bias distribution of modified nucleotides through coding, non-coding regions, promoter regions, genomic islands, and some other associations with GC content, GC skew in bacteria. So what we need as input files? We need basically 2 files, GFF file with modified nucleotides, and GBK file is genome sequence, annotated genome sequence. Where can we get these files? We suggest to use third-generation sequencing facilities like PacBio, for example, and PacBio create for us long reads and store all information about kinetics of base calling. This information is stored as initial BAM files. FASTQ files have no this information. So you have to request initial files, assemble reads into genome, then again align these reads against the genome, and program like IPD summary can analyze for you kinetics of base calling and statistically predict locations of modified nucleotides. So this is a long process, and problem is that with every new version of PacBio, they change a little bit format of data storage. You need always to use the latest version. But what is good news, many sequence providers like INCABA, for example, can do the job for you very nicely and provide you with a GenBank file and with a GFF file. GFF file is a text file with locations of modified nucleotides and context sequences and all statistical data is here. And our program first can analyze, show you specific nucleotides which were predicted as modified. This is usually adenine or cytosine methylation, but also it can show for you oxidized guanine, which also predicted by the same program. So this is dot plot presentation. What statisticals? Very important that we want to provide tools to do some statistics. And I can give you several examples, interesting examples. GATC methylation, it takes at both places. It's very common for many bacteria, for Enterobacteria, and it takes place at both strands of DNA. At A, adenine methylation, and T marks here, it's at opposite location, opposite strand, adenine, respectively. And this is palindrome. So it's always placed here. You can see, but what we found interesting, that here additional modified cytosine. It's very uncommon actually for methyltransferases to methylate at both on cytosine and adenine at the same time. But it was not found for dimethyltransferase. Dimethyltransferase is not associated with restriction modification systems. It's often methyltransferase, which involved to some extent, there is many publications and gene regulation. We found that there is another, I can show you. So another cytosine next to GATC, which also very often methylated. So what important? Our program can take a sequence of this canonical motif. This sequence can contain ambiguous nucleotides R, K, so because motifs are very often is semi-conserved. It can take positions of multiple locations in motif, which were expected to be methylated. We can instruct program to show motifs which are methylated at all positions. Or we can instruct program to show these motifs which methylated at least at one position. Or very important, we can instruct program to show for us sites which are supposed to be methylated but remain unmethylated. Sometimes it's very important information for us what is unmethylated sites. And here even more complex motif. This is only part of super palindrome with 4 locations. And you can see in statistics how this distributed frequency of these motifs in genomes and associations with coding, non-coding regions. Very interestingly, that these specific motifs methylated mostly in coding sequences. They're rare in non-coding, extremely rare in promoter regions. We believe that these complex motifs can be important for mRNA regulation, actually, because they can create these stem structures, not for level transcription, but post-transcriptionally. Another interesting example is methylation in Staphylococcus aureus. It's again complex motif with multiple N between semi-conserved 3 nucleotides. And this adenine methylation, this A and T at both strains. What is interesting, GVAG part always present, most commonly present on one replicore, on leading strand. Here, terminus of replication, it's, you can see smaller number. Opposite part, respectively, more common on lagging replicon. So there is bias between replicons. In addition to this, again, these patterns most common in coding sequences. But what is coding sequence? Gene is present in one strand. Opposite strand, we consider as non-coding. Yes. So this part, GVAG, always present inside of coding sequences, inside of genes. Respectively, opposite part is in non-coding sequences. So we can see this bias distribution between the replicors and between coding, non-coding sequences. Okay, another interesting approach is that we can compare distribution of methylated parts in chromosome and plasmids. In this case, we have one archaea, Allopherax vulcani. it has two patterns of methylation: it's cytosine and adenine. And what is interesting, cytosine methylation is abundant in plasmids, but relatively rare in chromosomes. And methyltransferase is present in plasmid. What is interesting, it targets specifically promoter regions. So it can be mechanisms how plasmid interact with host by targeting specifically promoter regions. And adenine methylation is common for chromosomes and some plasmids, but rare in another plasmid. If we want to do assembly from metagenomes, this approach also can be used. We very often create hybrid assemblies. And now we can use, because methylation patterns are very specific for individual clonal lines, for individual strains, we can distinguish between contigs generated from one sequence and another sequence, even they belong to similar organisms. Okay, program availability. You can find program at GitHub. There is a link. You can run program when you download it using command line. You can see here motif is set GATC, true, expected to be methylated, minus true at opposite strand. And you have to provide path to GFF, GBK files. If there is no arguments, program will show for you this menu, command line menu. You have to select which specific graph you want to create. And dependently on what you select, additional sets of attributes you can set here. And there is many arguments you can set, and you can see help file in GitHub, or if you type --help, it will show you detailed instruction of all arguments. Additionally, program available, there is a web interface. So you can get this access at this address and use, there is a smaller number of arguments are available, but you can analyze your files here. Program validation. Actually, this program, I worked in this program a long time ago, then students came to work, so it's already version 3, and some intermediate variants were tested in many projects, and there is some publications, 5 publications, where this program was validated. One latest publication is about interaction between archaea symbiont. You can see here, DPAN symbiont, very small, 500 nanometers, but it has a very reduced genome, but it has own restriction modification system. What we found, that in host organism, there is almost no methylation in these sites, but if we have host plus symbiont, we have multiple methylation at sites specific for symbiont. So through pores, they can exchange even big proteins like methyltransferases to prevent chromosome of host from a restriction enzyme of symbiont. Okay, a little bit of future directions. One new paper was accepted 1 week ago, where we consider interactions between methylation and gene level of expression of genes in different strains. We found out we use 4 strains of Staph aureus and show that they have the same genes but different patterns of expressions, very stable patterns of expressions, which are very difficult to change. And we believe it's associated with architecture of these genomes, and we use non-canonical methylation as to identify which genes are likely more expressed and less expressed. What we found that there are specific regions where presence or absence of motifs, methylated motifs, is negatively associated with a level of gene expression. Okay, so it's all done all in collaboration. We have 2 teams collaborating with us in Italy, in Kazakhstan, and we're waiting for collaboration with you. As Scott said, we need data, and we'll share with you Nobel Prize. So don't worry. Yes, thank you for your attention.
Thank you, Oleg. One burning question. We only have time for one burning question.
And the last slide that you showed before this was actually part of the question I was going to ask, and I'm curious in terms of visualizing this map, I know that very often we have the circularization of where the methylation sites are. Given the fact that bacteria reorganize the genetic instructions relative to the environment, do you have a way of placing those methylation sites on the operon map versus on the genome?
Okay, in this case, what we have here, this place locations of methylated sites relatively to start codon. And in this case, it doesn't matter where it's located. But you can see a drop of methylation in a non-coding part in all genes, not in specific in all genes. And more methylation is promoter regions, which are associated with regulation of these genes. So we can use with global map, or we can go to distribution of these methylated sites relatively to start codons of operons or some genes.